Good morning, this is Dr. Chakraborty. Today we'll explore one of the most elegant biochemical pathways in existence, glycolysis. Glucose is our body's primary fuel, but if we have too much glucose, then we risk diabetes, but without it, well, we wouldn't last long enough to complain. Let's see how our cells cleverly turn glucose into usable energy. Every cell runs on ATP, the molecule that pays for all cellular bills. Whenever ATP, ADP plus phosphate energy is released to power life, muscle contraction, nerve signaling, ion transport, where does ATP come from? Our favorite factories, glycolysis, TCA cycle, fatty acid oxidation, and ketone body oxidation. These pathways also mint NADH and FADH2, which later cash in for more ATP inside mitochondria. Think of NADH, FADH2 as rechargeable batteries. When they're full, you're energized. When they're empty, that's Monday morning reassessment. In bio, oxidation means losing high energy electrons, think extracting energy, and reduction means gaining them, think storing energy. Metabolic pathways that produce NADH, FADH2, ATP are oxidative. Those that spend them are reductive. Easy test. Did the step create NADH? Then energy was harvested. Glycolysis occurs in three acts: energy investment, 2 ATP, splitting, forming triose phosphates, energy generation, plus 4 ATP, plus 2 NADH, net aerobic, plus 2 ATP, plus 2 NADH per glucose. This is the on-ramp to mitochondrial metabolism. Pyruvate from glycolysis becomes acetyl-CoA, the ticket to the TCA cycle. Keep the storyline. We invest, we split, we profit, like a very predictable startup. What is acetyl-CoA? Acetyl-CoA is an acetyl group carried by coenzyme A, vitamin B5 derived. Acetyl is the only fuel the TCA cycle accepts, but coenzyme A has to do the delivery. It looks like a small piece of cheese stuck with a giant spoon. From glycolysis, pyruvate converts to acetyl-CoA plus CO2, pyruvate dehydrogenase in mitochondria. If oxygen is present, acetyl-CoA enters the TCA cycle. If not, we'll soon produce lactate. Before we dive into the details, let me give you 4 key points to keep in mind. First, glycolysis occurs in the cytosol. Second, the end product varies. It depends on whether oxygen is available and the type of cell involved. Third, several steps are irreversible. And fourth, glycolysis intermediates can branch off into other pathways for biosynthesis. So remember, glycolysis isn't just about energy production. It also provides building blocks for the cell. Why glycolysis? Glycolysis traps glucose in the cell and converts it to pyruvate, aerobic, or lactate, anaerobic. Major ATP bonanza comes later in TCA, etc., but glycolysis is required first. It also enables galactose and fructose metabolism and supplies intermediates for biomolecule synthesis. As an example, under hypoxia, e.g., ischemic tissues, lactate rises because cells lean harder on glycolysis. Glycolysis is like the campus shuttle. Not in Roseman, you might not end at your final destination, but you're not getting to TCA without it. When glucose enters the cell, glucose can enter glycolysis for energy production in every cell, or excess glucose can be stored as glycogen in the liver and muscles and produce biomolecules, e.g., ribose nucleotides, 2,3-BPG, RBC oxygen delivery, and amino acid backbones. Think of glucose as the Swiss Army knife, energy blade plus many handy tools. Where glycolysis happens and who relies on it, all cells have cytosolic glycolysis. Some cells must rely on it. RBCs and lens cornea cells have no mitochondria. They can't use TCA or burn fatty acids. So glycolysis is their lifeline. Brain has mitochondria but can't import dietary fatty acids due to the blood-brain barrier. It prioritizes glucose and, in fasting, ketones. This explains why hypoglycemia makes the brain unhappy very fast. Getting glucose in. GLUT transporters determine which cells receive glucose and when. GLUT1 widespread, high in BBB, RBC, fetal tissue, low KME 112 MNM, great for baseline uptake and hypoglycemia. GLUT2 liver, kidney, pancreatic V cells, very high KME 17 nanometers, only active in well-fed states, required for glycogen storage and insulin secretion. GLUT3 neurons, placenta, lowest km, wise KNME, brain first. GLUT4 muscle, adipose. Insulin regulates the activity. Very important in the context of understanding diabetes. Mnemonic. 1 is everywhere, 2 monitors plenty, 3 feeds the brain, 4 follows insulin to the door. GLUT logic plus hexokinase versus glucokinase. GLUT1-3 are low km guardians. They grab glucose even when blood levels are modest, supports CNS RBC. GLUT2 only ramps up when glucose is high, letting liver store and V cells sense and secrete insulin. Enzymology, hexokinase, most tissues, has low Km, is feedback inhibited by G6P, and prevents waste. Glucokinase, liver V cells, has high Km and high Vmax, isn't inhibited by G6P, perfect for post-meal surge handling and glycogen synthesis. Light humor, hexokinase is the frugal shopper. Glucokinase is the warehouse club member, buys big only when there's plenty. Energy investment phase to ATP. We phosphorylate glucose by hexokinase and glucokinase and trap it. Then in third step, fructose 6P, fructose 1,6 biphosphate. These steps consumes 2 ATPs. This spend now phase sets us up for payoff later. Then aldolase splits F1,6 BP into DHAP and GAP. Triose phosphate isomerase interconverts them so GAP proceeds. Energy payoff phase, plus 4 ATP plus 2 NADH. GAP dehydrogenase generates NADH while forming 1,3 BPG. Phosphoglycerate kinase yields the first ATPs, substrate level phosphorylation. After shuffling carbons, mutase, enolase, pyruvate kinase makes the final ATPs and pyruvate. Net per glucose, plus 2 ATP, 4 produced, 2 invested, and plus 2 NADH. With oxygen, NADH heads to mitochondria for big ATP. Without oxygen, pyruvate, lactate to regenerate NAD+, and keep glycolysis running. Practical line. When oxygen is late to the party, lactate volunteers as the designated driver for NAD+. Let's summarize the major concepts that we just discussed. Where does glycolysis happen? Cytosol, not in mitochondria. This is why cells without mitochondria can still do glycolysis. What does glycolysis produce? Net ATP gain is 2 ATP, spend 2, make 4, so net gain is 2. 2 NADH molecules. Key phrase is 2 ATP and 2 NADH under aerobic conditions. What is the major rate-limiting enzyme of glycolysis? Enzyme PFK, full name phosphofructokinase, is the main control point. Glucose transporter, GLUT4, is the insulin-responsive transporter. GLUT4 is insulin-dependent. Found in muscle and fat tissue. After you eat, insulin is released to mobilize GLUT4 to the cell surface, whereas GLUT2 serves as the glucose sensor. Km is the highest, roughly 17 millinits, found in liver and pancreatic V cells. Acts as a glucose sensor that responds to blood sugar levels. Remember, GLUT4 relates to insulin, and GLUT2 has the highest kmeans and serves as a glucose sensor. Hexokinase is present in all cells. Has low kinin, which means high affinity for glucose. Glucokinase is present in the liver and pancreatic beta cells only. Has high kinase, so it is only active when glucose is abundant. Allows liver to store excess glucose after meals. Finally, remember, RBCs rely entirely on glycolysis. Remember, RBCs will die without glucose and glycolysis as they have no mitochondria. Now you are ready to take a short quiz. Good luck and thanks for watching the video.